Thank you everyone for joining us today. This is a regular Friday seminar in the Open Source Neuro series, which is part of the Worldwide Science Initiative. This series is all about open tools and methods in neuroscience that we collect in our website, open-neuroscience.com. You can upload projects to the website and sign up to give a talk about your own projects using our forms. Please consider collaborating, share our content, and following us on Twitter. You will find all relevant links in the description below. Today, we have the pleasure of hosting Dr. Chinna Devarapu. Dr. Devarapu pursued a PhD in physics at the University of Exeter in the UK. After his PhD, he joined as a research fellow in the nanophotonics group at the University of St Andrews in the UK too. He is currently a Marie Curie Career Research Fellow at Munster Technological University. He loves low-cost hardware projects and all things open science, and his other research interests include silicon photonics, photonic crystals, synthetic biology, and biomedical devices. We will now listen to Dr. Devarapu's talk about the Mobilefuge. You can ask questions in the YouTube live chat, and at the end of the presentation, we will invite attendees to continue the discussions with the speaker in Zoom. Hello, Chinna, and thank you for being with us today. Please go ahead.
Hi. Thanks for the nice introduction, Celia. So I'll be going to share my screen now. Went to the last. Yeah. Hi everyone. Thanks for signing up for attending this talk. Today I'm going to talk about Mobilefuge. It's a low-cost, portable, open source 3D printed. One second, I would like to put the laser point. 3D printed centrifuge that can be used for purification of saliva samples for SARS-CoV-2 detection. This is the work I've done in collaboration with Mohni, Bhupati, and myself. I'm a Marie Curie Research Fellow at Munster Technological University, Ireland. This is the picture that represents the work which I'm going to present today. It's a centrifuge that you can plug into the smartphone, not quite literally, but I'll come back to that. My first foray into open source projects is through ODX project. Maybe some of you are familiar with this, where we hacked a fitness tracker to monitor the bacterial growth. And we try to monitor the many bacteria like E. coli and Streptococcus, all these. After doing this project, I was very motivated to try building many other biology tools, particularly based on smartphone. Why smartphone? Because there are already 3.8 billion smartphone users in the world, and mobile phones, I mean, not smartphone, but mobile phone users, if you see, they are 62% of the whole population has at least a mobile phone. There are many scientific devices already built based on the smartphone, like microscope, otoscope, retinoscope, or many other devices. You can find some in Open Neuroscience maybe. But there is no centrifuge based on smartphone. So I would like to build one. But coming to open source centrifuge designs, there are plenty of centrifuge designs, as I can show here. A paper fuse and a 3D fuse. The one that is based on a fidget spinner. This is a recent one from Manu Prakash Lab, a handy fuse. This is one based on, again, fidget spinner concept. It's a Dremel based one, and this one is based on a bicycle, and it is from a kitchen appliance, and it is based on a drone motor. So if there are so many centrifuges, why I need to build again? Motivation is from COVID-19, because during this pandemic, there are people suffering with COVID-19. They need to be tested to control the spread of the disease. And we need to isolate the people who are tested positive from the rest of the population to control it. Out of all the testing techniques, LAMP assays are can be cheap. They are very useful for the community level testing, and which requires frequent testing, like every day, for example, reopening of the colleges, something like that. They are more comfortable than RT-PCR if we use saliva as the testing fluid. But to conduct these lamp assays, there are two tools required: a centrifuge and a temperature source. Why centrifuge? Because if there are reports published that if if you don't purify the saliva sample, saliva as it is might contain many other contaminants, and that can hinder the lamp assay. So, and it will miss the low viral content. So still lamp assay can work, but only with the people who has high viral load. So to purify the saliva samples, a centrifuge is required. And Manu Prakash lab and some others in the Harvard University, they found that you don't need really very high powerful centrifuge. One can use low speed centrifuge. For example, this handy fuse was published uh, with a complete essay detailing how you can do low cost lamp assay using enzymes that can be had for less than $1. And this is the only device that required. I mean, for heat source one can use thermos. And recently there is a um, boiling water based test is also there for heating source. But for purification, a centrifuge is required. But even I showed many varieties of open source centrifuges. Let me review very briefly the limitations of current centrifuges. There are commercial ones, of course, one can have even for less than $100, but they require usually continuous power supply, and the high power ones are only available in centralized facilities. If a centrifuge is kind of acquired any damage, then it is hard to repair without sending it to the manufacturer. In research community and DIY community and maker community, there are many types of centrifuges exist. They can be broadly divided into electric based or human powered. In the electric based, they are mostly done with drone motors or hard drive motors because they can give high speed. But at the same time, they also require high power, usually 12 volts supply or high current around 1 amp. So this kind of speeds and current requirements raises the issue of safety. And to build these kind of centrifuges, one must know soldering and programming, things like, skills that are not very common. And they also require time to procure these components and to build them because they involve complex build procedures. On the other hand, the human powered centrifuges like handy fuse or fidget spinner based, they have a risk of splash or spill because they are not with any enclosure. It's difficult to put them in enclosure and operate them at the same time. And they might also cause repetitive strain injury, and you might not be able to use it for high throughput. For example, one would like to test hundreds of samples in a day, then it might not be a very comfortable thing to use. And another important thing is there is usually no speed control on these centrifuges. So the Mobilefuge is developed based on these limitations. So to address some of these limitations with a open source design. For that, I am based on the concept of Mobilefuge on these key observations. For example, there are more 3D printers than previously available. During this pandemic, we come to know this, how many people use 3D printers to print the face shields and all. And there are, as I mentioned before, there are many smartphones and mobile phones exist. So 5 volts power supply is kind of widely available. For example, if you want to use 5 volts and less than 1 amp, that's 5 watts. 5 watts is very easily available nowadays, and it's widely usable than any other voltage and current source. So based on that, since the lamp assays have this potential, let me build this centrifuge. And I will also show today, I also built a small temperature source based on these observations again. So what I would like to have in my centrifuge is, I would like to build with off-the-shelf parts, no custom parts. It should be portable, low cost, easy to fabricate, easy to operate, and can be powered from phones are, standard 5 volts USB port available in mobile phone charge, batteries, rechargeable batteries are computer USB sources. What I don't want is custom PCBs, no milling, soldering, programming, or access to grid power. Based on these concepts, I built the Mobilefuge. It just required a USB fan motor and a 3D printed rotor for a very basic way of getting it work. You just replace this fan blades with a 3D printed rotor, and it can rotate and it can give the centrifugation required. So here I would like to show the short video for a minute. So as you can see there, it is easy to make a centrifuge out of it. It's not a big deal, but the problem is you cannot use centrifuge like that. It is again will the same limitations apply, like safety, spilling, splashing, all these problems can come. So what I did is I developed an enclosure that is based on jar and lid model. So it consists of two lids and 3D printed rotor. We can put everything together like this, and it will be quite compact. So the importance of this jar and lid model is that you don't require special tools to build it. Once you have all the parts, building centrifuge is literally less than a minute, which I can show here. That's a OTG cable, if people are wondering what it is, so that you can power it with a smartphone. Okay, now it's good that it's working and it looks safe, but what about the speed and force? So it in many of the open source designs where you find the centrifuge built, but they never show the real calculations of the safety, they don't highlight at least the safety part or the testing with real samples. So I would like to overcome some of those with my project. So we need to estimate how much force we can generate with any centrifuge. So this force is given by this relative centrifugal force. It compares how much force or acceleration the centrifuge gives to the particles relative to the Earth's gravitational force. So you measure the force and divide it with the force of gravity, and this boils down to this equation. So if you know the speed and the distance, this is the distance from the center of the rotor, you can estimate how much force you can generate. For example, in our case, we have these 3 centimeters or 4.5 centimeters from here. That translates to RCF of 1800 to 2800. And we know from the literature that RCF of 600 is quite enough for the lamp essence. So we followed the same handy-fuse paper and try to separate the silica particles in water. That's called milk, glass milk solution. So within less than 1 minute, we can able to separate the water and silica particles. And not only that, we also show how to improve the stability. For example, we kept a smartphone, this is open source software running and measuring the acceleration on the same surface where the mobile fuse is kept. And this part of the graph is when mobile fuse is not switched on. As soon as the mobile fuse is switched on, you can see in this z direction, acceleration is kind of varying a lot, as you can see with this number. So to dampen this kind of vibrations, we can put a common furniture damping sheet, and that drastically reduces the oscillations. So we showed that it can be quite stable while operating. And now comes to the speed variation. As you can see here, we can connect a USB fan controller. These controllers available for less than $1 again, and we can put it through the same USB cable, and we can now vary the speed for whatever you want. It's limited, as I said, but we can still vary the speed. Following the same principle, I would like to also develop the LAMP device, although it is not focus of today's talk, but based on the same off-the-shelf components and portability, low cost and easy to fabricate and operate. We used a small circuit, this is called W1209. It's $1.5 if you would like to buy one, and using 3D printed heat block, 3D printed heat block and a cartridge to build one. So as you can see here, we made a 3D printed enclosure, and this is the place to put the tube with the saliva and primers. This MobileLAMP is realized with, as I said previously, W1209 circuit board. It is very widely used circuit board, mostly for incubation of eggs in the developing countries. It is based on a 20 cents microcontroller from ST Microelectronics, and it comes with a nice NTC thermistor, and along with a relay, and it can vary the temperatures from minus, it works in this range. It has the onboard programming switches and indicator LED, on the LED screen, and features to adjust the temperature and all these things. I don't want to focus here, but I would like to show where we are going after the microfuse. This is the few assembly pictures of the LAMP device, and this is the testing. Here, as you can see, I kept a thermistor here and plugged into a 3D printer circuit to check the temperature variations. As you can see, 65 degrees is quite stable, which is required for the LAMP SAS. And this is for another LAMP SA, which is being developed. Now we tested this device with at Cambridge University. Just today, some of the preliminary results came. It's Smith Hedge mostly doing the work in Jenny Mollay's group. Now these are the reagents before the LAMP assay, and after 30 minutes, the positive sample has the changes the color. This is with our heat block, our device, MobileLAMP device, and it is with the standard thermocycler. So in my view, the MobileFuge is a very great device. So it gives very simple to use centrifuge, stable, scalable, cost effective, open source, and low power. But more interestingly, it can be integrated along with the LAMP device with the heat source to make it as a one kit. Of course, it has its own challenges. For example, right now integration is not yet clear in many ways. For example, one can use magnetic separation, or one can use centrifuge separation, and the open source enzymes whatever have been published, we have tested our LAMP and MobileFuge devices with that reagents. So far we are using off the shelf, but in the future we would like to use that less than $1 costing reagents and see if we can scale it up, and testing with real virus particles, not just synthetic viral particles. So, but it also brings up the question, like, if we publish these things, and how we need to be how much we are liable for these results, and how, while especially while scaling up, just publishing might not be a problem, but if you would like to scale it up, how much we will be liable for example, the situation like this in COVID-19. Some places they don't have any access. For them it might be life saving, but at the same time we have to have some liability. We need to address these liability issues. So and all this project, both the Mobilefuge and MobileLAMP, both are all funded by myself. There is no external funding involved so far. So I don't know how good it will be to talk to the funding agencies, how much they can support. I mean, at this time I'm just developing in my spare time, so maybe when we would like to look for the scaling up, maybe we need to look this aspect, like how to get more funding so that we can scale it up and test it multiple labs. And at the same time, sparing time is money. So right now I'm doing this work in my spare time, but I would like to focus more on these focal science projects, and develop many open source hardware devices. So I would like to know how others in the community are doing it. So and how they are balancing particularly their original background, the deep science projects, with these frugal science projects. Maybe it's good to have the chart. Yeah, that's all from my side.
Thank you so much for that wonderful talk. Hopefully some of the people can join us in the Zoom meeting so they can discuss with you about this project.
Okay.
I was wondering whether you had reached out to any of the other projects you described before, such as Handyfuge or any of those, and how if you've interacted with them.
Yeah, I had a call with Manu Prakash. He reached out to me. He's the one who developed the Handyfuge. He mentioned that Centrifuge is going to be a next big, device, a critical device like his fold scope. So he would like to, make a network of people who are working on similar technologies. So I'm looking forward to join such kind of, any network that
Yeah, that
amplify
would be
the,
fantastic.
impact of this work. Yeah.
Yeah, it's so relevant nowadays. I was also wondering what other kind of assays you could tell us about that could be done with the Mobilefuge.
Um, SS in a sense, wherever you require DNA or RNA purification, for example, in the wild, one can use for example, there is a nice IGM project I have seen. They're trying to extract, plant cells to know the vine infection to the vine trees. Situation like that, wherever you need to extract the DNA RNA from these cells, one can use the Mobilefuge, because it's easy to take along with them, and they can just operate directly from the smartphone.
Yeah, exactly, and the low cost and ease of putting it all together, as you showed in your videos.
Yeah. So previously, for example, let me go back. Yeah. There are many excellent applications, demonstrated with the DIY or research grade centrifuges. All these can be easily applicable to the Mobilefuge, but some situations, some applications might require very high speed. There, the limitation of Mobilefuge, it cannot go for speeds more than 7,000 RPM.
Yeah, that was another thing. How I was thinking, because I can't see any questions just yet in the chat, so that's why I'm going off with all of mine, and Mattis, who is helping us in the chat today.
Yeah.
Our questions are these, and we were wondering what challenges, or whether you had already been seeing adoption and deployment of the Mobilefuge, and what challenges arise in the field. Let's put it that way.
In the field or in the building, I don't know, I didn't get your question, but in the field might be, 3D printing, it is not so fast, it's not a fast technique. maybe, for example, the entire print process of this took more than 12 hours. And as I shown here, these are different different types of rotor designs I experimented with it. One of the challenging part is this part. That's why I kept this picture as a representative, because the small hole to go into the fan is very critical to functioning of mobile fuse. So yeah, build process has its challenges. For example, as I said, low speed of 3D printing, but it is also good in a way that it can be injection molded, all these parts, if you would like to go high, scale it. And it required only few parts, like 4 parts. That standard lid and jar model tells that that is one of the highly injection molded part, because the jars and lids are used in many parts of the world. So maybe that challenge can be overcome if we can go with the injection molding.
Fantastic. Thank you so much for your presentation. I hope that you can reach the funding bodies and other collaborators to work on this further. There don't seem to be any questions at the time being, but we are all welcome to reach out to Dr. Devarapu on Twitter. And please join us next week on Friday at 15 UTC. Thank you.
Thank you.